Hello everyone, how are you? Let's take our second look at the instructions for life, and this time we're going to look at how those instructions get passed on, and that's by cell division. Cell division is how complex creatures grow and repair their bodies, and also make new cells. You began life as a baby, I assume, and you needed to get bigger, and so you needed to make new cells to do this. Cells do this by dividing in two, and they need to copy the genetic information inside and pass it on to each of the new cells. There are two processes that we use for cell division. The first is mitosis, which is normal cell division, replacing cells and healing and growing. Mitosis copies everything in the cell exactly, and it divides the cell into two identical daughter cells. The second type of cell division is meiosis, or meiosis. This prepares the genetic information for passing on to babies. It changes the information and makes new cells which have half the amount of genetic information. We can see then that mitosis creates diploid cells, but meiosis creates haploid cells. Both mitosis and meiosis are divided into phases, which we will talk about below. Let's start with one phase that is often overlooked: interphase. Cells spend most of their time not dividing. This is when they do all of the normal cell things that help to keep you alive, and this is called interphase. Before division starts and after it is over, all cells spend most of their time in interphase. Let's start with mitosis. And for both processes, I will start off by explaining it in words, then we will see it in pictures, and then we will talk about the phases, so you have plenty of chances to pick it up. First, a cell will copy its genetic information. We're not going to talk about how this happens, but it uses RNA as a kind of chemical copy and print machine. We end up with a total of 96 chromosomes inside the nucleus, which we now call chromatids. This process is called replication. The chromatids are connected into homologous pairs, and during cell division we call these pairs chromosomes. But after those chromatids are separated into new cell nuclei, then each chromatid goes back to being called a chromosome on its own. Yes, this naming system is kind of confusing, sorry. The chromosomes move to the center of the nucleus, and the chromatids are pulled apart from each other and dragged to the opposite sides of the nucleus. We now have a complete set of 46 chromosomes at each side. Now the nucleus divides into daughter nuclei, and then the cell itself divides into daughter cells. This process of division is called cytokinesis, which we will talk about in a moment. Let's look visually at mitosis. Here is a cell nucleus with some chromosomes added. We have only put a few in. Remember, there are 46 of these in each human cell nucleus. These chromosomes replicate to form chromatids, and those little X shapes are now called chromosomes, each made of two chromatids. The chromatids will migrate to the equator of the cell, the middle of the cell, and spindle fibers will form and attach to the chromatids. These spindle fibers are like small wires that attach to the chromatids, and they pull them apart, and the cell divides, and that way you have a complete set of genetic information in each daughter nucleus. And then the daughter nuclei divide into separate cells, and the daughter cells are exact copies of the parent cell. They have exactly the same genetic information. Each part of the process has its own name. The first phase is called prophase, where the replication takes place. And then we have metaphase as the chromatids prepare for division, and then anaphase as the chromatids are pulled apart, and finally telophase, which completes the cell division and produces the two identical daughter cells. Let's review the stages of mitosis again. We begin with prophase, when the chromosomes double themselves into paired chromatids. Then we have metaphase, where the chromosomes line up at the middle of the cell and the fibers form. In anaphase, the chromatids are pulled apart and the nucleus begins to divide. And then finally in telophase, the cell division completes and you end up with two diploid cells, each with two N chromosomes. Let's briefly again mention the process of cytokinesis. After the chromatids have been split, the cell has to form a new nucleus and split into two, and this requires a lot of things to be moved around inside the cell. So cytokinesis is a word that means cell movement, and it's the word we give to the process of the cell rearranging itself inside to prepare for the final division, and that final division into two cells is called cleavage. We will now move on to meiosis, which is quite a lot more complicated. Meiosis begins in the same way as mitosis. DNA will replicate and that will make chromatids, a total of 4 N chromatids in each cell, paired into 2 N chromosomes. Now comes the difference. After replication, the homologous chromosomes pair up and swap sections of DNA, and this is when the genetic information in your mother was changed to create the egg that would make you, and the genetic information in your father was changed to create the sperm that would make you. After the homologous pairs of chromosomes have swapped or recombined their genetic material, they are divided by cytokinesis into different daughter nuclei. All of the chromatids are now different from each other. Each nucleus after the first division has 46 chromatids in 23 pairs. This is the end of meiosis 1, but we still have paired chromatids instead of single chromosomes. Now there is another division, which is this time very similar to mitosis. The chromatids line up in the center and are pulled apart again into different nuclei, and this is the process of meiosis 2. We now end up with 4 haploid daughter nuclei. Again, let's see that visually. Here is our cell nucleus, this time preparing for meiosis. The chromosomes replicate to form chromatids, and then the homologous chromosomes meet up and swap parts of their code, so you'll now see that we have each chromatid being a different color, and all of them are different from the original nucleus. The nucleus now has 4 N chromatids, and it divides into 2 nuclei with 2 N chromatids each. The next stage is that the chromatids line up at the center and have a mitotic division, and at that point we end up with 4 daughter cells. Each cell is a haploid cell with N chromosomes, and the chromosomes are all different colors from each other and from the parent cell. These are called gametes, which we mentioned above, and these are the special sex cells that go into eggs and sperm. Pop quiz: how many chromosomes and chromatids are there in each stage of meiosis? Pause the video and make a quick guess. But let's look at the top left cell nucleus first. This is a normal cell. It has 46 chromosomes and 0 chromatids. Then, in the top middle, after the replication has taken place, there are still 46 chromosomes, but there are now 92 chromatids. After the first division, each of the daughter nuclei has 23 chromosomes and 46 chromatids. And then the chromatids are separated by mitosis, and so in the final stage, each cell has 23 chromosomes and 0 chromatids, because what we were calling chromatids are now called chromosomes again. Just like mitosis, we use the same names for meiosis, but there is a slightly different process. Prophase 1 is when the chromosomes double and they also pair up into tetrads, so each tetrad is 2 chromosomes and 4 chromatids. Tetra is a prefix that means 4. Genetic recombination starts here, which is when the genetic material is swapped between chromosomes, and it continues in metaphase 1, where genetic recombination completes and you end up with chromatids that are all completely different from each other. In anaphase 1, the homologous tetrads are now pulled apart to opposite sides of the cell, and in telophase 1, you divide into two haploid cells with two N chromatids. Remember, these are haploid cells because they have only N chromosomes, even though they have two N chromatids. Then we start prophase 2. The chromatids prepare for the second division, and metaphase 2 is when the chromosomes line up at the center of the cell, just like in mitosis. Anaphase 2 is a mitotic division when the chromatids are pulled apart to opposite sides of the nucleus. And then finally, we have telophase 2, when the new nuclei form and the chromosomes are finally divided into two haploid cells, each with N chromosomes. We have gone through both processes several times here, but don't worry, you can always watch the video again if you aren't sure. Let's finish off by comparing mitosis and meiosis. The purpose of mitosis is growth and repair. The purpose of meiosis is to produce sex cells. Mitosis takes place in all cells except gametes, and meiosis takes place only in the reproductive organs where gametes are made. Mitosis produces 2 daughter cells, but meiosis produces 4 gametes. Mitosis produces daughter cells with the same number of chromosomes as in the parent cells, but meiosis creates cells with only half as many chromosomes as the parent cells. Mitosis produces no variations in the genetic code, usually, although mutations can happen. But basically, the daughter cells from mitosis are exactly the same as from the parent. This is not the same as in meiosis, when the daughters mix genetic material from the parent cells to create new genetic combinations in the gametes. Finally, there is no variation between the daughter cells in mitosis. They are all identical clones of each other. But in meiosis, all 4 of the gametes are genetically different. That's all we'll go through here, and I hope you now have a little bit better understanding of cell division. If not, you can always watch the video again, or maybe you can find another video that will help you to understand better. You can watch Hank Green again on Crash Course Biology. He has more detailed explanations of mitosis and meiosis. They are not designed for students whose second language is English, but they are excellent and detailed introductions. But thank you for watching this video. I hope you found it interesting. If you have, why not like the video or subscribe to our channel? And you could possibly pass it on to a friend who is also studying cell division with you. Bye for now.